So now that we understand kind of the structure function relationship with lipids, we can start to talk more about the functional aspect. So when we put lipids into lipid bilayers, they have fluid properties, and this is important for the way that our cells work. Lipids can move in two different ways. They can do transverse diffusion. This has the half-time of days. Why might there be a half-time of days for a lipid that's up here? Oops, excuse me. For a lipid that's up at the top of the bilayer to then kind of jump to the bottom of the bilayer. Why would this lipid not really want to jump to the other side of the bilayer? Well, think about this. The kind of head group of this lipid is charged with the phosphate and maybe has other types of polarity depending on what's going on with that X group. How likely is it for this charged polar group to travel through the non-polar lipid tails of the bilayer? Probably not going to happen. So the most common type of movement for lipids is lateral diffusion, where this lipid just kind of will move laterally through the leaflet. This happens really fast. These lipids are just zooming around. And we can use simulations to show us just how fast these lipids are moving. So I want to show you this video. One thing you want to look at, how much are these lipids moving? Are they kind of moving back and forth through the membrane? But the other thing that I'm using this video to show you is, again, another example of the hydrophobic effect. You'll see these lipid bilayers form spontaneously, and this is going to be driven by the entropy of water. So let's go ahead and start this video. In this video, what you'll be looking at are the green kind of sticks with blobs. Those are your lipids. So watch what happens over time to the green bits. You see that at first, kind of there's these separate chunks of green all moving around, right? You can see the lipid tails kind of waggling, some of the lipids kind of jumping around. But now it looks like these three separate chunks of green have started to form together, looking kind of like a bent lipid bilayer. And you can see that the green tails are all stuck in the middle, whereas the head groups are all facing these kind of gray water molecules that are bouncing around. And now as the video continues towards this end, you'll see that the bilayer is almost perfectly formed. It's flattened out, these lipids are bustling, they're still moving, kind of jumping around all over the place. Pretty cool stuff. So that's what's going on with lipids in a bilayer. But what else is there besides the phospholipids that we just saw in this simulation? Well, there are other lipid-based membrane components. Let me get my screen right real quick. There are other lipid-based membrane components. Of course, I already mentioned one of them that's super important besides phospholipids: cholesterol. Cholesterol is really key. It's important because it broadens membrane fluidity. It makes membranes more fluid because we have that cholesterol kind of sticking in there, getting in the way of some of those lipid tails, disrupting some of those nice London dispersion forces. So it makes membranes more fluid, but also because cholesterol has this four-ring structure, it does add a little bit of structural integrity through its own rigidity. Now, when you think about the bilayer, this is a very small point. When you think about the bilayer, just know it's not fully uniform. There are things called lipid rafts, where like certain types of lipids tend to cluster together, but that isn't important for the sake of this course. Just kind of know that the bilayer is not uniform. And just to kind of hammer home this idea of structure function, the breakdown of lipids is important to physiology. There are many enzymes, some that are native to our bodies, some that are not, that take lipids and perform some chemistry on them to change them. One example of this is phospholipase A2, which is found in snake and bee venom. What happens is this enzyme, pictured here on the right, kind of takes the lipid into itself. So this protein holds onto the lipid and then performs a hydrolysis reaction where it chops off one of the lipid tails, the one at the carbon 2 position. The product is something called a lysophospholipid, which is a strong detergent, and we'll find out why on the next slide. So this is why we need snake venom antidotes, right? When we start messing with our lipid bilayer, we start disintegrating our lipid bilayer. That's not going to be good for the fate of our cells. So what exactly is happening with this snake venom? And let's understand the structure-function relationship. Okay, so here's kind of my simplified lipid that I've been drawing for you. This is a phospholipid because we're going to be worried about what's happening to our lipid bilayers. So this enzyme in snake venom comes in and performs a hydrolysis reaction at the C2 carbon. This results in the phospholipid releasing that lipid tail, and the reaction results in a hydroxyl group at this point. But the other lipid tail remains, and what got chopped off was a fatty acid. So what's the structure-function relationship here, folks? We went from this lipid that is a phospholipid with two tails to a lipid now that is a phospholipid with a single tail. The van der Waals radius has changed, and it's gone from something that is mostly cylindrical to a van der Waals radius that is mostly conical. Now, if we think way back to the slide where I showed you the different structures that fatty acids make compared to bilayers, you can see that changing a phospholipid in this way is going to result in a structural change for the lipid bilayer. And this is why snake venom is so bad. Up next, folks, we're going to talk about the last but important type of membrane component, proteins.